Hi, we are going to talk today about how molecules move across the cell membrane. So I'm going to present here. Hopefully this will work well. Let me have a pen. Okay, so here we go. Turn down this sound for now. So hopefully you remember from physics, the second law of thermodynamics. This isn't just about physics. These same laws govern how things happen in biology as well. And as a reminder, it is that the universe tends toward more disorder or entropy. So if you see in this picture here, we have these molecules of dye that are in high concentration on the left side and no concentration on the right side. And they're in an aqueous solution. So there's water on both sides, and you could actually say there's more water on the right side and less water on the left side. And what's going to happen is that the molecules of dye are going to spread out due to the second law of thermodynamics until they've reached some sort of equilibrium. An important thing to note here is that there are holes in this membrane that can fit those molecules of the dye. If the holes were smaller and the molecules of the dye couldn't fit through, then you could never have equilibrium in terms of the molecules of dye because they would not be able to move down their concentration gradient. So if you look, you have high concentration here, low concentration here, okay? And the way that this works with diffusion is that molecules will move down their concentration gradients from an area of high concentration to an area of low concentration. Okay, so here's just a little animation, very high tech, showing diffusion. And diffusion is the movement of molecules from high concentration to low concentration. This is also known as passive transport because it does not require any energy in the form of ATP. You're going to learn a lot about ATP this year, and ATP is basically the energy currency in our bodies, in all biological systems. So we convert glucose to ATP that then we use in other places. However, we don't have to use ATP in order for diffusion to occur. It happens passively, so it does not need any input of energy for this to happen. Imagine if someone walks into a room and in the door they start spraying perfume or Lysol. The molecules coming out of the canister of Lysol are going to spread throughout the room until they're evenly dispersed, and then they've reached equilibrium. At that point, they might be such a small concentration, you might not even smell them at the far corner of the room, but they're still there, and they will continue to move for like all eternity, I suppose. When we have the movement of water, there's a special word for that, and it's osmosis. Osmosis is the movement of water down its concentration gradient. So here in this animation, you can see the water molecules are these little guys. They look like little flies or something. And you have molecules of, let's say, protein in the green. Note that the protein molecules are too large to pass through the membrane. And so the protein molecules can't move. So there's more on the right than on the left. When this starts, you have more water on the left. The water will continue to move over to the right down its concentration gradient, trying to balance out the concentration of those two, of the water and the molecules, right? So you can't get the molecules over, but you could dilute it with the water so that the concentration is equal on both sides. Potentially. Sometimes you can't. So osmosis is the diffusion of water. Okay, so you learned about the cell membrane and its structure and how it separates the outside from the inside, and that it's selectively permeable due to whatever channels are in it and the nature of the membrane. So the question is, can it be an impenetrable boundary? Well, that would be very problematic if nothing could get in or out. We need things to be able to get in and out. We need nutrients to get in. We need waste to get out. We need oxygen in. We need carbon dioxide out. So it's very important that things can move in and out of these cells. So what do we want in and what do we want out? What we want in is food or nutrients. So typically like in the form of glucose for the cell to use to make ATP. Carbohydrates can be the food, sugars, which are sugars, proteins, even lipids. We want amino acids in so that we can build our proteins. Okay. We also want salts because salts are an important part of homeostasis. We want oxygen and we want water. What's got to come out? So waste products. Okay. When we do cellular respiration, the waste product is carbon dioxide. All right. We also have a waste product of ammonia because we are like a land-based mammal. We secrete or excrete our ammonia in the form of urea. So that is carried in our blood, goes to the kidneys, and then we urinate the urea out of our bodies. Other organisms, for instance, marine or freshwater fish, things like that, can just get rid of their ammonia without having to convert it to urea. We can't. Ammonia is like too toxic for us, but they have enough water around to be able to deal with that. So ammonia's got to come out. Salts also will go out. Carbon dioxide needs to get out. And water. So water goes in and out all the time. Okay. And our cells are going to make things. Our cells are going to make proteins, for instance, to be used elsewhere. Our cells are going to make all sorts of stuff. They're little factories. So whatever they make has to be able to get out of the cell. Okay. So what can get straight through? I want you to revisit the idea of what makes up the membrane. So remember, we have these hydrophilic heads. Okay, that's the PO4. And we have these hydrophobic tails. So what can get through? Well, lipids can actually go straight through. So they are able to get from the outside of the cell through the hydrophobic interior and to the inside of the cell. So this is actually interesting in medicine when they're trying to make medicine that can like cross barriers, like the blood barrier, there's blood-brain barrier, and you want medicines to be able to get inside of cells, they will actually like pharmacologically make them hydrophobic so that they can cross the cell membrane. Sugars cannot go straight through. They need a transport channel. Amino acids cannot go straight through. All right. Water, we used to think could go straight through, but then we learned that they have to go through teeny tiny pores called aquaporins that I mentioned the other day. And the reason why it can't go straight through is because of the hydrophobic interior here. That's blocking it. Salts that are inside the cell cannot go straight through because they're polar. If they're polar, right, salts are polar, like NaCl, Na+, Cl-, that's polar, it's hydrophilic. It can't get through that hydrophobic interior. NH3 cannot get through. So polar molecules can't go through, ions can't go through, and large molecules can't go through straight away. But what we're not showing here, and maybe it's on a future slide, carbon dioxide and oxygen can get in and out, no problem. Okay? They can go straight through because they are nonpolar. So protein channels are the way that things can get through the membrane, and they are specific to whatever is traveling through. So they're specific to a particular amino acid. They're specific to a certain type of sugar. So you have a ton of these. Okay, so that's how sugars are going to get in. That's how amino acids are going to get in. And water going through those aquaporins. Salts can get out that way. Ammonia, which is usually from the breakdown of amino acids, that can get out. So you have these channels. So this is called facilitated diffusion. Facilitated diffusion is different than just plain old diffusion because it needs a facilitator. The facilitator is the protein channel. So think of it as the helper. That's what a facilitator is. It's a helper. So it needs a helper protein in order for molecules to get in and out who are too big or that are polar and can't traverse the nonpolar interior of the membrane. So that's what the channels do. Again, because it's diffusion, you don't need energy for this. So here, in this case, if you count these up, we have like 1, 2, 3, 4, 5, 6 molecules. I guess maybe this, yeah, we have 6 molecules of, let's say this is glucose over here. We only have 1 over here. So this must be a glucose channel, and these molecules are going to move through the glucose channel to try and establish equilibrium on both sides of the membrane. So that does not take energy. You don't have to input ATP, but you do need a protein helper, so it's facilitated diffusion. Okay. Let's see. Active transport is different. Active transport, when I want you to think active, that means it requires energy. When you're active, you use energy. So active transport requires energy in the form of ATP. And the reason it requires energy is because you're going against the concentration gradient. You're actually trying to push more molecules into a space. And this is going to be super important when we learn about how different, like your whole, your whole nervous system works because of active transport and the sodium potassium pump that I'll talk about in more detail. So sometimes cells need to move molecules against the concentration gradient from an area of low concentration to high. So it's sort of backwards. I like to show this video whenever I am doing this. Let me pause. Okay, so I want to show you this video that I like to use every year for sort of to show you the analogy of what happens with active transport. So I want you to imagine that these people who are trying to get into a Japanese subway are molecules of sodium, and they are getting pushed against their concentration gradient into the subway. So take a look. So apparently in Japan, I don't know what city this is, but they have some overcrowding issues. And see these gentlemen with the hats? Their job is to push more people into the subway. Can you imagine? I would be so claustrophobic. Oh my gosh. Now they gotta shove their clothes in. Look at that. Oh, I don't understand what happens if you need to get out and you're in the middle. How do you get out? All right, off the subway goes. These people just shoved inside. Oh. So I like that animation or video because I think it shows what's happening in cells that are pushing something against the concentration gradient. That takes energy. So the guys who are the pushers, so think of them almost like they're the transport protein, they are using energy to shove people into that space that already has a ton of people in it. I want you to think about what would happen when those subway doors open at the next stop. All right? What would happen when those subway doors open at the next stop? I'm going to have you write down what you think would happen, and we'll talk about it in class. All right. So cells might need to move molecules against the concentration gradient. And what happens with these proteins is that you have a shape change, or sometimes called a conformation change. So the shape, if you see this, it's open on one side and then it changes shape and releases molecules into the other side. In order to change shape, you need to use ATP. And we'll discuss this a lot this whole year about how ATP, when you transfer a phosphate group from ATP onto a protein, it causes the protein to change shape. And that change in shape allows the protein to do whatever it's going to do. So it's going to move this molecule, for instance. If you see the words that it's a something pump in terms of membrane transport, that means it is active transport. Anything that's a pump takes energy. The same way like a real pump would take energy. So a pump takes energy. The energy is in the form of ATP. All right. So what you're going to end up doing if this is a pump is more and more of these are going to go through this pump until you have a ton over here and none over here. And then you're just going to keep pushing them and pushing them and pushing them until you've sort of stockpiled up these molecules. It takes ATP for that to happen. All right. So I want to talk in detail about the sodium-potassium pump. The sodium-potassium pump is one of the most important proteins in your body. It is a protein that pumps sodium in one direction and potassium in the other. What it does is it pumps out 3 sodiums and it pumps in 2 potassiums into the cell. So when you look at a cell, I'm going to try and draw a cell here. When you look at a cell and you're pumping out 3 sodiums. Look at this handwriting. That is a positive ion. That's a cation. I'm pumping out 3. This protein, this is not a picture of the protein, it's a picture of the cell. The same protein is going to pump in 2 potassiums, 2K+, which is also a cation. So why does this matter? Because it sets up a voltage inside all cells. Cells have a voltage, which is a change in sort of like the electricity, you could say, a change between outside and in. Every cell is about negative 70 millivolts because the inside is negative compared to the outside. Why? Because of the sodium-potassium pump. I want you to envision this. So if you give me, all right, you're inside the cell, I'm outside the cell. If you give me $3 and I give you 2, and you give me $3 and I give you 2, we're giving each other something positive, a cation. We're giving each other something positive, but you're giving me more than I'm giving you. So what's going to happen to you in the long term? I'm going to have you answer that question. So after some rounds of this, what's going to happen to you compared to me? All right, that's why we end up with the outside of the cell more positive than the inside, and that creates this voltage. This voltage is important for your entire nervous system to work, and I'll talk about that in detail in class about how the sodium-potassium pump creates this voltage. 25% of all the ATP that you make, so when you eat, 25% of the food you're putting into your body that's going to go make ATP is used to power the sodium-potassium pump. That's how important it is. And in your brain, it's like, or nerve cells, it's 70% of the ATP. So the sodium-potassium pump is really important. Very interesting little fun fact here. Maybe this will be in trivia. There's a chemical called wabane, and that is the chemical that's on like Amazonian poison tree darts, like the into your neck, you're dead. All right, the way that wabane works, and it's a natural substance from a tree or something. The way it works is it actually shuts down the sodium-potassium pump. If you don't have a sodium-potassium pump, you don't have a nervous system. If you don't have a nervous system, you're dead. And so it happens like this. And the reason you die, if you get hit by a poison tree dart in the Amazonian jungle, is because you no longer have a sodium potassium pumps. And so you don't have a nervous system. I want to show you this animation. Let's see. Ooh, discard. Let me go here and show you this one. This is showing the sodium potassium pump. So I am going to start the animation. All right. So let me start it from the beginning. Okay, here we go. I'm going to begin again. 3 sodiums, those are the red dots, enter this protein pump. So this circle here, I don't know if you can see my mouse, this is a protein pump, and it can fit sodium on that left side, okay? Then what happens, here comes ATP. This green molecule that has the 3 little dots on it, that's an ATP molecule. ATP is adenosine triphosphate. There are 3 phosphate groups on it. The ATP is going to transfer one of those phosphates to the protein, to the pump. Watch what happens. When it does that, now see the little green dot is still there. It phosphorylated the protein. By phosphorylating it, it makes it change shape. And so now it changed shape. It was like this, and now it went like this. And now these sodiums can now leave because the protein is a different shape. So what you'll see here, sodiums go out, potassiums can now fit into their side of the molecule, and that's the right side. The phosphate group falls off, and now it goes back, it snaps back to its original shape, and the phosphates will enter the cell. And this will go on and on like this, bing, 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 and it takes ATP to make it change its shape. When it gets phosphorylated, it changes its shape, 3 sodiums out, 2 potassiums in. And that's what creates the voltage inside of cells. All right. So going back to here, this is just showing you that same thing, just kind of in steps. So sodium binds, here's the ATP. Where's my pen? Let me get that to work. Maybe it won't. Okay, I don't know if you can see this glowing thing, but the ATP transfers a phosphate group to this protein. The protein changes shape, expels the sodium, then potassium binds, the phosphate group falls off, and then it returns to its original conformation, dumping the potassiums inside. So here we have something called antiport. So you have 2 sodiums going in and 1 amino acid coming out. Let's see if I can show that again. See the 2 sodiums in, 1 amino acid out. So sometimes you will have 2 things being moved, like the sodium potassium pump, and an antiport means they're going in different directions. Okay? Here, you have symport, where they're moving in the same direction. And notice you have 2 amino acids and sodium going in the same direction. So that would be a symport. Sym means same, anti means opposite. So we're transporting opposite ways or in the same direction. So this is just sort of a review of the different types of transport. We have simple diffusion, all right. Facilitated diffusion, where you need a helper molecule, a protein channel, and then we have active transport, okay? So that requires ATP. Okay. This is a nice little graphic for you. Like the explosion of ATP there. The last thing I want to talk about is how we transport large things that are too big to go through a protein channel. They could be proteins, all right? They could be bigger than the protein channels. How do we get those things into a cell? How do we get them out? So for instance, your white blood cells are always, I talked about how they're always like touching your cells and figuring out what's supposed to be there, what isn't. Well, if your white blood cell encounters a bacterium, for instance, it's actually going to engulf that bacterium, bring it inside the cell, fuse it with a lysosome inside the cell, and destroy it. That's how white blood cells work. Okay? So it does that by what we call bulk transport, meaning big stuff. So we use vesicles and vacuoles to do this. Endocytosis means bringing something inside. So what I was just talking about with a macrophage, which is a white blood cell, bringing inside a bacterium to destroy it. So there's two different types of endocytosis. Phagocytosis, that's where the cell is eating something solid, like a bacterium. Pinocytosis is where cell drinking, where it's bringing in liquid, like a big glob of liquid. I always think of like Pinot Noir is liquid. So cell drinking is pinocytosis, cell eating is phagocytosis. These are two sort of subcategories of endocytosis. Exocytosis, think about exit, that's where the cell is expelling something. So after the macrophage breaks down the bacterium, it's going to have all these pieces of stuff from the bacteria. It can then expel it back out of the cell, and other cells could use those parts to make other things. So this picture here is showing exocytosis, where you have a vesicle. Maybe the cell made some proteins, all right? You got to get those out of the cell because they're going to be used somewhere else. How do you do that? Well, the cell that has the proteins in a vesicle, and we'll talk about how proteins are made and they're in vesicles, that vesicle will actually fuse with the cell membrane and just sort of push it out. All right, and now the vesicle, which was a double layer little organelle, is now part of the cell membrane. So the cell membrane is always like folding in and making a little vesicle and pushing out, and the vesicle becomes incorporated into the cell membrane. So here's endocytosis. So that's like a bacteria coming into a cell and then fusing with a lysosome for digestion. Pinocytosis is the liquid. And receptor-mediated endocytosis. That's where there's an actual receptor for a particular molecule that allows it to then bring it inside. And when we start talking about cell communication, this will become more clear. All right, that's it. Thank you for listening. I hope it wasn't too torturous, and I'll see you in class.